G'day, good evening. I hope you're all doing really, really well. This is a medical cannabis program. I'll be talking about some
G'day, good evening. I hope
plant tissue culture. Does it come through that loud? Holy mackerel. I'm talking about, cannabis, safe use of cannabis and its ways, and today is going to be on plant tissue culture. I do respect the law, and I hope you respect the law too. So today is going to be pretty decent. There's a lot of things I've prepared for that. I'll just have to get some of the stuff up on the screen and show you. It's because the end result is those nice trichomes, and inevitably trichome farmers. See what we can pull out of it. But today is a few different things. So I'm just slowly preparing. While I'm preparing, actually, say hello, g'day, Colin. Hola, amigos, ¿cómo estás? Raptor, g'day, Raptor. Nice to see you, mate. That's good. Yes, today is right up your alley. It's not real fungi orientated, but it's going to be tissue culture. You might be able to pick up some cool things out of it. Hello, Jono, who cares? Over there in Thailand. Sure, bro. Nice to see you, mate. Hey, cool. Brews in the house. All right, today this is the slide thing that I made up for it. There. So a bit of there is a bit of fungi on, huh? Fungi? It's not today's. Remove. I usually get the slides ready before. I didn't today, and what a mistake that was. That was last week's one, the fungi. Yes. I was thinking, there's not much today on fungi, but today, I tell you what, it's the 1 year anniversary of the show. So I'll be doing these lives for 1 year now, and I've been enjoying it. It's been good, and thank you to all you out there, all you cool people who are helping me. So I appreciate it. People like Supreme Grape as well. G'day, Supreme Grape. So this is a little bit better. So this is the slide for today. So there's tissue culture. We're going through a hang of a lot of stuff about that. There's different studies I'll show you, different preparations, different home lab setups you can do, different techniques, medium and especially recipes. I've found it very hard in the past to get recipes because it's a bit of a secret thing. But yeah, I've got quite a few different recipes from different papers to show you. And then the microbeads or synthetic seeds, that's these little things in the corner here. I've made a different video on that. You can go and watch on how to do it and the ins and outs and stuff. And it's 1 year anniversary, yay. And a Q&A, of course. It's always a Q&A. I've got my tablet going so I can see if there's any questions that pop up. So just put the little question mark and I can see them to try to get to them straight away. Supreme Great. Congratulations, CC. We are everything you do for us. Thanks for the support, mate. Appreciate that. Yes, we've all decided to do this. Home medics, good afternoon. How are you? Tissue culture. So first things, I got some rad news. Rad news. You wouldn't believe what Australia's done. I'm going to share this. It's nearly a world first. It's yeah, and I wouldn't have thought it would have happened to legalize such full-on drugs. But Australia has approved psilocybin and MDMA for therapeutic use. It got approved just under 24 hours ago. So there you go. So you just got to go to your authorized prescriber, the same as medical cannabis, and you can get some MDMA or some psilocybin. That's full on. So you can grab the LSD or the Molly and get some action. And I can really tell you what, I really, really support psilocybin. I've used it extensively under medical conditions and had great results from it. But I only support cubensis. So I only support cubensis only. Yeah, and the reason because of that, because once you dry them, they'll shrivel up, and then you dry them, and then you can have your dose by weight. It doesn't go off the amount of potency that's in them. So when you snap them, you'll see the little blue dye that comes up. That's the reactive agent. So you know that they're got their properties in them. And then, actually, I wasn't going on about that. This is just the news. Yay for Australia. There you go. Forbes magazine. Ooh, look at that. Even celebrates Australia's good work. It's going to help so many people, dead set. What it does for people that don't know is psilocybin, used correctly, it resets you. It gives you, your body can have a detox when it goes through those processes, but for your brain to have a detox, what the hell do people do? They don't know, but I'll tell you what I do, psilocybin. Maybe once every 6 or 12 months, or when you need to have, after you've had an event or something like that, you can have just, I'm not going to tell you the dosage rates and stuff like that, but I'll have, say, that amount, and that will just get me, I'll put aside an afternoon and sweep as, and it sort of resets it. And then the next day, anyway, this is just news, so I'm not going to concentrate on that one bit. Other rad news is Luxembourg, they have votes to legalise cannabis as well. But the poor folk, I think it was saying something about 3 grams. Yeah, here. Buying and possessing more than 3 grams could give you jail time. So it's got a long way to go, but it's the start. Just like Australia's doing it, it's a start. So good on you, Luxembourg. And there was someone else actually that did some legalisation. Oh yeah, Ukraine's president says legalising medical cannabis can help people impact from the trauma of the war. Yes, as well as many other things. So that's really cool. Really, really cool. So we'll see if there's any stuff's being said. Actually, cheers, everyone. I'm in charge of learning this. Wow, how the universe works. Okay. So, all right, I'll get back to sharing the screen. What was up to? The top things. Tissue culture media. No, that's some studies we're going to go through soon. Oh, that's the wrong one. We'll be getting to that one later. That's my personal tissue culture file, which I'll show some stuff in that. But these are the slides I've got to go through today. This was pretty cool. Let's watch a little video. This is tissue culture automation. So they've worked out how you can automate it now. So you can make plants and just pump them out and keep pumping out. So they've written this program, and this all does it itself. The benefits of the automation is it's in sterile conditions. And that's one of the hardest things to do in tissue culture, because you can grow plant cells every 15 or 16 hours, they might divide, but bacteria and fungi can divide in minutes. So that's just so brilliant. So that's really the half of it. More than half of it is the sterilization thing, because you're just making media and then putting the stuff in the media with its right temperature. Is the next is to complete the task. So it's that was really cool. Here's another video or videos. What's this one showing? I can't remember. Oh, this is just a rad future on how drones are going to be picking fruit. This is how drones pick fruit. Where's the big screen thing? Hand-picked. Just so, they do everything. I thought, no, this is going to be some sort of joke, but they just do everything, and it's really nice and gentle as it wants to be treated. It's so cool. So soon, drones will be doing that. So that's another thing that people can do. Another thing for the Canna jobs will be, if you're into your automation, you can start working towards automation for cannabis, for tissue culture possibly. I'll go through a few little jobs, entrepreneurships in cannabis soon. Righto, the little videos. All right, back to the start. Yeah, so this is congratulations to Australia. Legalising MDMA and psilocybin is now legal. Congratulations. That's today's. Oh, and I showed a few weeks ago talking about drying, and this, I saw this picture and I thought I just got to show this because this is just really, it impressed me about the drying. So it is a commercial setup, but they've used coat hangers to their benefit, and you can see the amount of air gaps in between. There's quite a lot of air gaps. And you remember from the other ones, people bunch them up too much. And that's what gives you your mold and your fungus growth. So this is just, I was really happy with this. That's really going to reduce the amount of spores that can germinate because they need their right activity water level. And it's not going to get up to 0.7 or above, 14% moisture in the buds. So right in the center. So if you were to spike it and it says 14% above, there's a chance that Saccharomyces cerevisiae can germinate. And I like this one. This is I can relate it to tissue culture because I thought, wow, they're trying to make more space. How can we fit more tennis balls inside of a blimmin' container? And this clever person, they answered the question. Look at that. Well done. But they're not usable. But it's a similar thing with cannabis. What can we do? Can we shrink it down? Well, tissue culture is. It just does exactly that. This is kind of later on, but because we're here now, one of the best plant places or a real successful spot to grab your cells from would be a flowering plant, because here it says explants. Explants means what the host plant is. It's an explant. So explants isolated during flowering period regenerate more easily in vitro than during resting. So that's this is from my tissue class. Can't remember one of the classes it was. Been through that one. There we go. There's a nice little, just a little quick slide of how it happens fast. So plant tissue culture is common and is in many other crops and often used to breed seeds and grow whole plants from clones. It's used to multiply cells so fast, and so many other things you can do in tissue culture, which I'll get into the fine tuning later on. You can de-virus things, you can pick selective breeding, you can manipulate genes, you can make homozygosity instant in one generation. There's a few cool things you can do from manipulating this in a very tiny little space. So first you just cut them up. You take your cuttings from wherever you want. Usually a meristem is, if you haven't done it before, get some of the meristems from the tips, from your rams and your shams. your root apical meristem has right at the tip there, so you cut the tip off, or you cut where the nodes are. They have cells that have a chance to divide very fast. It's called totipotency, is the name of the plant's name of the process if the plant is suitable for tissue culture. And cannabis is extremely totipotent. So after you cut it up, then you want to multiply it. So you'll have to, you sterilize it first, and then you want to multiply it in your different medium. And then you change the medium after you form a little globular chunk into your shoots or your roots by you putting in your cytokinins or your auxin favoring media. And that's it. And really, truly, that's it. So the whole hardest bit is the media and the sterilization. Thank you for those people. This is my little button that's underneath for the people, $5 of support. So I appreciate the people that have done that. Thank you very much. I don't get anything. So $80 also is, that's for about a dozen odd of my private lectures, playlists. In my playlists of my YouTube, I have thousands of private lectures that are related to plant, soil science and microbiology. And you can merely just type in your problem. It'll come up under there and you can watch some lectures on it from university professors and doctors. $150 is for a 20-minute video call and a novelty gift like a chart or something that I've made. $300 is for a weekly video call, more so for a company getting started or for a person who wants my educated opinion, needing help with doctors or their path that they need to take. Can of jobs. What's the can of jobs? This is can of art. So if you're an artist, look how nice that looks. I'm sure some people want to have a look at that. Oh. This is for chefs or for catering folk. So they've gone and put together a can of cookbook. So there's probably not too many out there. Or you could actually, or even get the can of cookbook and do some parties or weekend activities for people. Now that it's legal, check your, legal people in your areas. This is related to legal folk. It's nearly legal everywhere now, so it's great. This is, I was impressed with what it is, but when you're a legal area, if you've got options, you can do this as well. Create these wonderful dab swirls. So that's just dab. And then they've gone and put keef over the top of them. So they've rolled the scoob, the joint, and then they put the dab swirl. So they would have like held it up and swirled it around and then sprinkled it, back sprinkled it. And yeah, that'll get you to where you want. This is good because it's medical cannabis and it's related to art and just it's the way the world's going. So if you're talented, you can get and create quite a few of these things and probably get some good money for that. Entrepreneurships. And for those folk who are talented with drinks or fermentation, this is to show the charts of how well it's done just of recent. The amount of money that's come back in from cannabis drinks just in California in 2023, it's outstanding. And this is the average price per drink. So look at this one, $22 a can per can. And it's yeah. But still there's just money to be made. So just giving the cool folk out there options to make money out of the stuff that we really love. And I just put this in because this is rad. Look at this. Flying cars, George Jetson, meet George Jetson. Here he is. Yeah, it's cool. 2024, Suzuki are going to do it. Onya Suzuki, that's Japanese technology. That's insane. So they were changing it from a 2-car and they changed it to a 3-car. So they just altered it. But just excellent. That's not cannabis related. Here we go with the home setups. So this is just one example of a home setup you can do. I like the setup. Mine's got 2 little holes in it. Should have probably taken the phone anyway. Because here it hurts with the sides. See how you get in and you end up cutting yourself on this. But if you've got 2 holes, cut the holes decently so your hands can move up and down and then it's good. So you can create conditions like this. Just one example of going through. Oh yeah, there's a question. I see a question mark. Automation, yes, crazy. Kane Corso says, have you any experience with canna embryo rescues? Embryo rescues? Yeah, yep. Di-haploidization. When pulling the anthers, using that in a culture and culturing that, rescuing that, that's about what I've done that. And that's about it though. But I'll go through the different ways that you can do that. Excellent question. I like people who are interested. Thank you, Kane Corso. John who cares says, I will sign up. So CC, I need your help. Okay, mate. Thank you. Appreciate your support. Back to the screening. Yeah, there's quite a few different ways, Kane Corso, that I'll go through. Because I've done 3 university courses so far on it, on tissue culture, on plant cells and tissue cultures, and it will, there's various techniques in there on how to do different processes. I just summarized it before by saying it was the sterility in here. I just summarized it by saying it was the sterile and then the medium, because there's multiple ways that you can culture the cells, multiply a cell. Yeah. That's something. This is a little bit of another cool slide. Just shows using the floral parts, because remember the floral parts had a higher chance in reproducing faster. So you go and you dissect a few little bits off it. And these are called the bracts, perigonal bracts or calyxes. Actually, do you want a quick session on taxonomy, cannabis parts? Write in chat if you do. So I've said it a few times. Let me know. Then you go down here. So you take the parts, you'd sterilize them, induce, put them in your media. They start growing. Then you change the media to whatever you want to form them like before. And bang, you acclimate them and get them ready for outside. Cool as. And why should you tissue culture? Why shouldn't we just clone? What's wrong with that? Well, here's a good comparison of tissue culture versus cloning. So The space needed for 1,000 cuttings, this is more so for commercial, but it's a lot of space for clones, and tissue culture is not. Clones per person per day, you might get 200 or 250. Actually, you get a hang of a lot more, but that's what this study says. And in tissue culture, you can do a lot more because it's just how fast can you subculture a callus chunk? So how fast can you cut up a piece of potato and divide it, really? Multiplication rate per month. So you might only get 1 or 2 from cloning, but you get 4 or 5 from tissue culture. And the cost is way cheaper, way cheaper. The clone multiplication in 3-month cycle is a lot more because you can process more. Cleanliness, yes, well, it maintains disease and pest free in the, so if you're in an area that's very, like in Thailand, for instance, that's very airborne pollution might be rampant and there might be harmful parasites flying around, more so a reason to do tissue culture cultivation for your next generation coming through. And the vigor, sometimes, well, yes, there's a chance you're going to stress them. So the stresses are reduced in tissue culture. So therefore, it's probably going to remain as it should when the stresses it's going to have to induce its immune system. And the yield, well, 10 to 20% natural increase in cannabinoid content because of the vigor and health. Okay, that's what they reckon. I think it depends, yeah, how it grows after that. You know, they don't flower them in tissue culture, so that's, I don't really agree with that. Estimated clone production, estimated revenue. Yeah, so that's a good comparison anyway. Oh, that's when legalize them. Oh, that's the home TC setup. Sweet. All right, done all them. Is there another? I think there's another question in there. I think so. Oh no, there's Canna Corsos. Yeah, embryo rescue. Get to the different processes later on, mate. Thank you for your interest, though. Appreciate that. Cheers, bro. Haircut. I like it. The problem in Thailand. Jono, who cares, says he lives in Thailand, and the problem in Thailand is the pest, and obviously this will stop the issue. Yes. And also, if you've got a pest riddled plant, you can do a meristem culture and remove the virus out of it and have a clean plant afterwards. Certain person in BC did that to Death Bubba for a fee of $7.5 grand. Thank you. Where's the slides? I'm going to share first. Present. Share screen. We'll go back into, I'll go to those studies later on up top. So here, I'll finish off with today's stuff. CBD drinks, that Sky thing. Today, microbeads. Why didn't that come up before? This is the microbeads. So you mix sodium alginate and calcium chloride together and you can get these. I made a video on it if you want to know. So you just mix them up and it's a synthetic seed and they store well and they're really hardy. You can't squish them. It's cool. And yeah, this was a study done on it to see how cool they were and to see what effectiveness they had. So to be honest, I can't remember. And they don't end up yet to heat them. You have to heat them to get them to delaminate like that. Culture period. What's this? Cumulative regrowth rate. So there's a high, yeah, you get a high regrowth rate if it's in the right conditions that it would like. Not sure what I was up to. Oh yeah, that's right, the home setup. Oh yeah, here's another one. And this is a cool show how it goes in. If you first goes in the lab, then it's into your different, depends what media you put in. So the plant cell needs 5 things to rejuvenate. And if you get all those 5 things right, you're going to get this rejuvenation. And agar uses medium, like Raptor, he'll be using his agar because he does heaps of that. He's agar loving, or you can use gelin. Is another cool thing. The advantage with gelin is that it's a lot clearer. And you can tell if you're growing any types of bacteria or anything else is culturing. And then it goes down, and these culture vessels cost a bit of money. So the cheaper version is Chinese containers. Get the ones that are suitable for the microwave. No worries. And yeah, put them on your little dish. They don't require much light. You just got to leave them for 3 to 4 weeks, start doing their thing. Then after that, that's it. What's the next one? In that, oh yeah, this. Oh, let's just show you the when to change the medium, I suppose, because it's definitely rooted. I can't remember what this experiment was, because that you can see this one's got a lot better lateral roots than this one. So its primary roots both decent, but this one's got, so this would have been maybe a gas was, but no, actually I'm not going to hypothesize what the experiment was. And I like this one. Dirty water doesn't stop plants from growing, so don't let negative words stop your process. So if you're in the dumps or anything like that, just shrug on, try and do your thing. That's last. That's so technical. I'm going to get up to this one, tissue culture. Actually, I should show the best one first. This is a study done on it, and they found which is the best medium to use. I'm going to show everyone a basic one, because we all haven't got top dollar. We're not really interested in all these fancy words and all that sort of stuff. We just want to blim and do it. And we want some words, some action. So this is a really rad broken down version and recipes on how to do it straightforward and simply. It was so appreciative. It's just excellent. So you just got to prepare your sterile place, really, because this is for home. So you want to prepare an area which is out of wind. You don't want carpet because that can put spores into the air. You want to try and get maybe a bathroom where it's tiled or its floor is glazed type thing, and there is little air, and then sterilize it. Maybe spray some bleach or spray some. There's a good cool recipe in here to make your your ethanol, your bleach spray. You don't use bleach though, but you can do it a bit better with your. Anyway, we'll get to that. So it's just all about making your right place. So I showed that before. Oh, I got rid of it. Remember that little container upside down? Cut 2 holes in a tote, 2 holes, and there's your container. Sterile, spray, get it all ready. Next. Open you get your explant. So you want to work out, where's some more photos here to show? Get your explant. So what am I actually doing? Which part do I want to culture? Do I want to do a meristem culture? Well, if you want to duplicate it, the easiest way is a meristem. So just cut a node where these flowers connect, just in case the bracts haven't got the requirements that you want. You can guarantee when it's already in its flowering cycle, so it's already induced that way, and it's got the flowering hormones going through it. So cut a node off, which a node, I'm sure you'll know what a node is, where the petiole meets the main plant stem. So where it attaches on. So where you cut it off. So, and Google meristem if need be, right in the center of it. So if you just cut a little tiny bit, fingernail size you want, and then instead of that has meristem or parenchyma cells, and those cells multiply very, very easily. Back to it. That's where you want to find your explant. So after you found that, you'll go and sterilize it. So you'd generally put 70% alcohol. The reason why you don't use 100% is because it evaporates too fast, and there's a chance that it might suck in all bacteria and things like that. So 70% has a little bit of water in it, so it just maintains its sterility for a little bit longer. Oh, cool, here it is. There you go. So you have that, but I recommend do it with 2. You'll find you won't get cut. But there's many ways to do things, just like growing. You can do it all your different ways. So yeah, get that, put some holes in it, put a little working light. I use a UVC light, so I put it in the back corner, and you've got to have a candle too. So you have your candle in there to knock it, so when you're doing your cutting, everything here will be sterile, but still things can get in. And if you have failures with this, you're going to have to put a little fan at the back with a HEPA on it. A HEPA intake, so it'll blow in through here and out. Then you'll have luck. But usually don't go to that extreme first. You don't have to buy that. So with the UVC light and a burning candle, around the candle when it's burnt, there's no microbes or pathogens, or it's free of everything because it's just burnt very close. So if you were to do a culture in that area, so when you're cutting or things, do it around there, and you're guaranteed nothing can contaminate it or mix with it and then quickly dip it into your medium. You can go, I'll go through this slowly just in case you want to read it later on, because it is a pretty cool read. Preparing the alcohol. This is rad. This is so easily done, so cheaply done. So if you haven't got access to ethanol, if you can't go to Walmart, Walmart sells it. They've got the rad already made up, 70%. It's real cheap too, like $10. You can make it out of metho. So get your metho, pour a third out, and refill with water. There's your 70% ethanol. Put it in this refillable aluminium hairspray bottle, $2 from a reject shop. No worries. There you go. And preparing the media stock, this is the hardest bit, really. What's Rap to say? The irony of needing more water in order to kill and dehydrate the microbes. Yeah, it's funny, isn't it? How does that work? These are the hardest things, is the stock solutions. So I haven't written down my media. I can. This is for the cytokinins, and naphthalene abscissic acid is the oxen. So this is if you want to make a. Actually, I'm going to. Depends what media you want to make at the start, but you don't want one of these solutions. You want a callus making medium. The reason being because the callus is easier to work with, and then you can divide it after that. You can subculture. I think we're going to get into, I'm going to tissue culture. All right. What do we got here in my plant classes? Where's the callus chunk? Is it there? Yes. See, here's the callus. So that's what you want to make a media to do this first. Instead of making the roots or the shoots, I suggest doing it that way. And then you can choose what you want to do, because that callus chunk is rad. Out of that, that's cells that haven't made up their mind what to do. So all that you've got to do is give them the right hormone, and then it'll tell it what to do. So you give it the right cytokinins, and that produces the shoots, or you give it the right amount of auxin, and that produces the roots. So it depends which way you want to drive it, but I'd suggest just doing it that way first. These are the recipes. To cut a long story short with recipes too, you can go and get MS medium, Murasagi's and Scoob medium. Get one with, what is it? I get a pre-made, so I don't do all this. I add my own hormones though, but I get MS medium with added vitamins and sucrose. So it adds a good amount of sucrose and vitamins, because you only need 5 things, and that MS medium pretty much has 4 of them. So it's basically done for you, and it's cheap. So that's M and S medium. S for Skooj, Skooge, Murasagi, and Skooj medium. They're the blokes or the people that made up that blend, and it's a blend that works pretty good. And then just get your cytokinins and your auxin. And kinetin, what do I use? I think I use kinetin too, but I use IAA as well as NAA, naphthalene abscisic acid, and IAA, indyl abscisic acid. So that's the hardest bits, the medium. Materials, well, I'll just pause it there so you can go and read that. Distilled water, yep. Microwave, you can use your sterilising instead of the pressure cooker, because if you don't want to buy a pressure cooker, you can still do it in the microwave. Just make sure it bubbles for about 3 or 4 minutes, and that means it's got to the heat, unless you're at altitude. Pickling jars, you can, instead of them, you can use your Chinese dishes, the round ones, because they're a bit cheaper, a bit easier to stack. Don't need the culture tubes or the clone bottles. So there are a few ways you can get a little bit cheaper out of this, just to break it down. This is the stuff you can't find anywhere, or it's hard to get a reliable one. You might get one that's people have made up but they haven't had success with. So these things are very hard, because it's all cultivar dependent, because you find different ones need different things. Like as you've seen in the past, when you've grown, like Pink Death Bubba was a potassium pig, and then Newcomb used to be a nitrogen pig. So things like that, you can tell from your cultivars which ones you can alter to get to the right outcome. So this is, yeah, some general formulas. Hey, where's that? I'll give you a bit of a help here. When it's talking up here before, it was saying about the right formula mix for, see this, dissolve 0.2 into 200 ml. Well, how do you get 0.2 of a gram if you don't have scales or even powder scales, or 0.02 of a gram? Because with you get different quality strengths of your powders, of your oxygen or your hormones. You can get like lab 91 and 95, and there's 97 strength and all this. So how do you blend them up to give the outcome that you want? So you go, don't buy them, go to the rooting hormone powder, look at their dilution rate, and you'll find theirs is about 0.2, or it's in that vicinity, 0.05, or it's something, it's really, it's pretty close to this vicinity. So that'll help you a lot with that as well. I should check questions. There's some. I have to go to work. I want to stay. Oh yeah, Jono. No worries, mate. You can catch it later. Hey Dave, how you going? Nice to see ya. Raptor, yes, just make sure to allow steam. Oh, okay, Raptor says that. Yep, cool. PC are ideal, but not absolutely necessary. Okay, thanks, Raptor. Appreciate these people, their experience as well. It's rad. There's a rooting medium. So after you've subcultured, you'd want to put an oxygen heavy medium in there. So that's a solution for that. Rinse the water, making slopes. It's just a color overview. Very good. Why can't I scroll? This is an overview. So if you just want to read through that, you can just go through a bit of an overview later on. You'll be happy. But it's pretty straightforward. Just collecting plant material. So after you've cut I didn't really go through this at the start. I just skipped it. So you cut it off, and then you've got to dip it in a solution. So you want to make sure it's sterile. And then out of that solution, you think, how do I work this? So you've got to take it out of that solution into another solution again, and then out of that solution into another solution, just to make sure that when it goes through the air, that it's not picking up things and it's getting rid of stuff. Like if you were to use bleach, or if you were to use ethanol or something that's in that solution, you don't want to transform that into your culture medium. So you'd have to go through a few solutions to get rid of it if you're going to sterilize it somehow. My hands are hurt. Can you go like that? Yes, I can. Right. So there's how they do theirs. It's a technique, so you can read through later. I have to do this, huh? 13. Transform to a root and medium. So after you've got your callus chunk, you want to transfer it. So the calluses, how long do they take to grow? Well, the plant cell takes about 15, 16 hours to multiply in favorable conditions. So after that cell multiplies, it's got to redo that again. So then times it by that. So you're up to 1 day minimum just to make 2 cells that you've got to see under a microscope. So to get to something that's the size of you want about sort of my middle fingernail or something like that. Or no, that's pretty big. Pinky nail. So your pinky nail. That's not very accurate. 1 centimeter or about half an inch. When it grows to something as big as that, that's quite big, and you know you can subdivide it. And that will take approximately about 3 weeks. In favorable conditions, mind you, you might get better. You might get in 2.5 weeks, but you might also get it in 4 weeks if it's a bit, the temperature's not kept at what it wants, for instance. Because remember, this is, treat it like a newborn baby. If you hear a newborn baby whinging because the medium's wrong, the temperature, or the medium, which is the rooting material or stuff, or the temperature's wrong or something like that, they will cry. So you've got to treat it like that, and then you'll have better luck. Preparing to plant out. That's pretty, I'll put this in just because it's good reading anyway. It's a good basic understanding when they don't talk about technical stuff on what to do, because it shouldn't be made technical, because it's not. There you go, you completed tissue culture. Sweet. So that's a bit of a step on process on how to do it. Now what was I up to? Oh, that's right. Week 27. We're into this. How to isolate. Good, that's all done. Is there any? I'll just be polite and see if there's any questions or anything. Dave said g'day. G'day, Dave. Oh, yeah, and Raptors made his good suggestions on his experience. Thank you. Okay, get back to, oh no, I've been through all that. So the top bit, so I have to share again. We'll go through what some papers have said and some experiments now on what different lights do and different media does. So this is from a company that does it. So these are the few things. The laminar flow hood, that's a device that tote that we make. So you don't need the laminar flow hood if you can successfully do it under your conditions, which as you see, laminar flow hoods, get into it. So this is what they say you need. Your ingredients, get it pH'd, pressure cooker, laminar flow hood, your hormones, and then put it into your culturing vessel. So meristem, this is the reason why, it eliminates viruses if you do a meristem culture. So if you get it into I tried to zoom then, into the meristem, it hasn't had a chance to, the phloem and the xylem haven't had a chance to grow in there yet, meaning it hasn't transported all of the other cells and material from the plant. So if you culture in that little bit there, though it has not picked up the virus, so to speak. So then you just duplicate it. You've still got the same DNA, the same 32-odd thousand genes that the cannabis plant originally had, but it doesn't have that virus in it. So then you just put that into the callus chunk, and bang, you produced a plant without that virus. And truthfully, it's that easy. What's technical there? You can nearly just read that. Just sterilization, and then I've sort of covered them bits. Pathogen remedial using meristems. So that was what we were talking about before. It's not that technical. See, they haven't even got a fancy looking microscope. Look at their stereo microscope. It's not even that fancy. So you can just do it. You don't even need a microscope really to do it under, because you can just see where the little bit of the node is, and you just cut around the node. Is there a picture for that? No. Where there is a bloody picture. I'm going to find one. I haven't got much on preparation, just on the growth of it. No, it's not even one. It's a nice one of the callus chunk. Where's the little explants that have been cut and ready for sterilization photo? Thank you. Start. I can't see it there. That's disappointing. What about? So, no, that's pollen culture. Embryo rescue. All right. Sorry about that. I was trying to find it. Now I'm just lost to where I was. Gender screening. Okay. Well, they put it through various tests and just to get some gender screening out of it. A few different ways you can gender screen. And long-term storage, synthetic seeds. Oh yeah, that was those synthetic seeds, those round-looking microbeads. Okay, that was for that. Cannabis synthetic seeds. This is a study on it to see how effective they are in cannabis, how well it works, and it works well. Here's the name of that medium, if you want to know the spelling of it. Murasage and Skoog medium. So you buy that with the vitamins plus sucrose already in it, and you don't need much afterwards. Whoa, and there's a bit of something special for you because you're watching. I'll just share this little thing here. That's hard to find. Yes. Microbead recipe. I didn't even share that, I think, on the video. Wow. Where's my microbeads while we're talking about it? There's the video I made on it. Look at that, just little caviar. It's amazing. There you go. That's with the cannabis. That was a leaf culture I did inside of it. So if you want to do like the meristem culture before, you just cut off your different explant location. Depends where you wanted it to do. I was trying that time with the leaves, tree spirit. Back to this study. Has there been any tests? That's pretty annoying. Yes, just allow. So we'll go down. Here we go. This is what before I showed this screen and said, what is it for? I wasn't sure. It's the impact of light exposure. So it depends how much light exposure. We should always treat it with the same 18 on 6 off when you're trying to, when it's regenerating, or even in the color stage, because you can do it in the dark, and it's etiolated, it's called, but it hasn't gone green. To build up the green chlorophyll pigments, you add the light to it, and I really suggest doing that. Really low light fluoro tubes. I'll run through, probably a good time now to show some setups while we're there. Microbeads. Sculpture. Here we go. Some rad setups. So you see how much lights they've got under there. One light per rack, and it's suffice. Just one again. No heating pad, because they would have regulated the whole room. This is lighting, changing the different spectrums. There's a cool study I'll go through later on showing the different spectrums of the lights. That's their setup. The fluoro LEDs, they might have needed 2 instead of the 1 fluoro in the middle. These ones, see the gap distance is a lot higher, so they needed 2 fluoros. So it wasn't quite designed as well as they could have had it. Efficiency-wise, this has got, look how many fluoros put inside for here. There's way too many because there's a height difference, because that's a changeable shelf at the back of it. This is more utilising the space a lot better, but they've got vertical lights, so it's just different again. Here's one only, and you can see there's bits of dark patches. But for this cultivar, it was fine. And all you're doing in veg, you're just growing it very, very you want it lightly growing, and you can see that's just perfect. It's loving what it's doing. So from one light under all of them, what's this one? Oh, this is a cool chamber. That's, yeah, nice, a whole room that's just set up and not really spaced utilized as well, but maybe they could just they could go double height. Back to that. Right, that was a good thing for the showing different setups. How's just while I'm looking at the other screen too, is the connection coming through all right? Today I just tried the high definition and it's looking a bit blurry on my screen. Can you please get back to me if it's blurry? It's coming through blurry on my other screen, which isn't good. Far out. Raptor said some stuff. Nice synthetic seeds and propagation in bioreactors are the most fascinating aspects of tissue culture for me. Yeah, and it's fun as, mate. Just grab those 2 ingredients and you mix at a certain thing. You mix the, put one into the other, and it just happens. And then it's all sterile already. So you put them in your Petri and it just grows. And you start to see, depends what medium you put in it heavily, but it starts to throw shoots or roots out of it. It's fun. Yeah, can you guys let me know if, girls, let me know if the stream's blurry, please, because I adjusted the settings for high definition this time, which I don't know if I should have. It's still looking blurry on my other screen. How could I change it even if I did? Looks good to me. I do adjust. Oh, okay, good. Thanks for the feedback, mate. Appreciate it. Okay, good. I can continue. It's hard here. When I was up to the study, we're into a few studies now. This is all the fun stuff. So that was how to do it, like a real basic on how to do it. I really could elaborate on how to do it, but I just want to get into the experiments bit because I could talk forever on it, and we've nearly been going for an hour. So there's so many different experiments now I've got to show on the outcomes on what to do if you do this. Like, what's this one show? Oh, this is the microbeads to show how totipotent they are, and they work really well. That's the growth rates, different media. The vigor of dark stored syn seeds. So you can see that they've done this test here comparing dark of sprouting them in the dark to sprouting them with light. So even if you're a little bit electricity hurt or something along those lines, it's possible you can leave them in the dark for the first week at least to 10 days. And you'll start to see little blobs of white, sort of green, creamy stuff growing. And then you introduce it to a bit of light. That's possible too. It still works all right. So that's what that showed, Brad. Oh, there's a germination of the syn seeds after they're planted out. Well, that's pretty obvious how they'll go after that, as long as because once it's got the root hairs, that can pick up different minerals and vitamins that it needs and start processing them. So it should power on. Once you've got roots in, you just change media. Explant preparations, encapsulation matrix. That's to do this synthetic seed matrix, encapsulation seeds, that's what they're called. I've done a few other ones that I've gone extensively into this encapsulation methods because there was my class in plant science covered this and went right into detail. So I've already covered that. So if you want to know more about that, go back to I've done a live on it on tissue culture, and maybe check out the other couple of videos that I've done on tissue culture. Actually, I did a world's first on tissue culture. Did you know that? Yes, and it still hasn't been, I've put on the page, if anybody's done this prior, please let me know and I'll update my details. Science is cool how it changes all the time. So this study just proved that it works well. 100% regrowth rate after 150 days. So they could adjust. So to get speed on that, they could have, oh, here you go. Ultimately, it had a balanced matrix. So if they could go and adjust light, temperature, media, all those types of things to speed that up. So that's, I think it was just an overall, just to prove that it works fine. Put on. Here's a cool study that's on the use of LEDs in tissue culture. So how does it work? What does it do? Why do we want red? Why do we want blue? So I've found red, it is not good for rooting. It kind of inhibits it, which is not good. So I've played around with the different colors a little bit. So I tend to go with blue, and I've found blue around, I think it's a 440 nanometer, to be precise. That's the spectrum it comes out of the LEDs. It gives an inducing of the cytokinin and of a cytokinin type expression, and throws branches out quite well. Here's some other different effects. Studies of different red on different spectrums in the plants, just to see what the outcomes were. So that's from my experience. I just wouldn't really use the reds at the start. Maybe you've got cultivars that do. There's all sorts of mutants as well that have like ethylene insensitive mutants. There's ones that inhibit different spectrums as well, mutants. So you've got to really see what works with your genetics. See here, they've kept the room, so they wouldn't have gone and sterilized their media and their thing as much because their room's sterile. So see, he's wearing a root, but look, he's got hair there, so that drops off. So they're not sterile. So I don't know. I like keeping them in their own, making them sterile first, and then keep them in their containers. But mind you, this is a grow out room. So yeah, sorry to interrupt. this is showing the different lights on the different outcomes on how they work, the effects of different light spectrums for pest management now. So this was pretty cool, just to showing the difference on how in the UV spectrum on what it does. So if you've got a tendency to go and produce a lot of these types of certain type of bacteria or fungi, you can go and introduce different spectrums and see what works best for you. Because see, some spectrums work, and some in the blue, for instance, work really well. Yeah, look, the blue, they attract a lot of things. Attraction, and it can be used as a repellent for no things and some response, but attraction. So it's, yeah, depends what you want to do in your area. But that's how the lights can infect with your microbial growth too. And look, once it gets right up into the greens, because you know green isn't uptaked by much through photosynthesis, so it's a highly attractive one for the bugs. So if you want to try and get a lot of bugs coming in, maybe grab a few green spectrums into your setups too. And then once you go right down to the infrared, which you put down, you use for putting into sleep for the Emerson cycle, it has no response at all. So the insects aren't interested when it's in the morning or in the evening, just in the middle of the day with the yellow and in the blue. So that was pretty fascinating. So you can take that into account when you're, yeah, look at that, lime green, pure attraction, citrus yellow, pure attraction. So it's something that hasn't really spoken about much. The effect of different light spectra on control growth of microbial populations. Does it increase them? Does it inhibit them? What happens? So red is promising light for photodynamic therapy that can effectively inhibit bacteria, yeast, and microcrustaceans. That's red. Blue can inhibit grey mold. So botrytis, if you've got that growing on your plants, this is maybe, or if you're susceptible at that time, you first see a little sign of it, maybe put some blue on. Grab a sodium metal halide, CMH, no, metal hydride. What's the old school ones? The old school lights. The old school ones that they had a very blue spectrum in it, that some people do. You could go and use that as an example, but this is talking about LEDs. But it's more the spectrum that they're after. So it's just, that's what this is talking about. So yeah, like here, see the blue, 405. I got a 440 nanometer LED. So if I was to use that, I could have similar to complete inactivation of Streptococcus, those round ones under the microscope. So it's just different effects have cool things. Bacillus, Pseudomonas. And remember I was talking about last week how you can identify what genus you have by a few colors. So conclusion, we don't need a conclusion. So yeah, that was a good little interesting study to do with the LED lights, the spectrums, I suppose, on tissue culture. Here's some advances in tissue culture. When was this written? 2021. It's not real current. So what's this going to really say? Righto. Okay, got nothing to discuss there. This is where this is the chemical structures and the properties of, like, the medical effects of the terpenes and cannabinoids. I'm not going to go through them because there's heaps. Like, I remember there's heaps. Do you know it's an interesting, while we're going past turpentine, where is it? Turpentine, terpenol. I think it's that one, terpenol. The reason terpenes were terpene. The reason they were named terpenes is because, when was it? A while ago, can't remember. But the fellow that discovered it, it was because it was a smell coming out of terpenol, that terpene, and that's what they were originally named from. because it would have been turpentine back in the 1800s. And, well, I got a smell. And terps originally come from that. So in vitro, hairy root cultures. There's so many different cultures. I'll get into them soon, the different types of cultures you can do. Somatic embryogenesis or organogenesis. So you're going to involve with producing embryos and breeding type thing through di-haploidization, as an example. Or are you just going to do it by multiplying cells over here and producing organs with organogenesis? So I went through that way before to produce the callus, and then after that you mix to get what you want. And embryos is a lot different because you've got to wait for the action to happen. I'll go through some of them. Even though I have covered a lot of embryogenesis, embryo rescue things in the past with my tissue culture, that other tissue culture one. What's this? These are the studies in cannabis done with different genotypes, different parts of the plants. So what part of the explant were they using? So like were they using the calyxes, which is the bract, or are they using the root apical meristem? What happened? Did they put it into what type of morphogenic response did they want? The media they use, which this is so important, mate. See how they only play around with it's only the hormones, because you've got to get if you get the minerals right, remember the cell only needs 5 things, and 4 of them are taken up with the MS medium just about. So then you get your hormones. So that's why these are only altered with the hormones. So you just got to change your hormones. And it's that simple. Where's that chart? I'm going to show that chart. I'm still sharing. I've got to find it. There's a rad chart which shows you how much oxygen to put in. Actually, it might even be in here. There it is. So organogenesis. So if you want heaps of roots, put in heaps of oxygen, no cytokinin. The opposite, if you want heaps of shoots, put in heaps of cytokinin and no oxygen. And then if you want a callus, you have to put in both about the same. That's it. Plus MS medium and your what type of agar or gelin or type of media for the bottom, and sterility, and bang, you've got reproduction of cells. Where are we? So yeah, I'll go through slowly on these on these recipes, because you can come back and try it. Depends what cultivar you've got. It's a different medium. It's awesome. There's so many recipes. For me to have this, when I was starting this, I don't know, when I tried a few years ago, it's just so beneficial, because you just don't know. People just don't want to tell you, or they might lie, because that doesn't work. I don't get reproduction off that. Oh, you sure? Was this right? Did you have a good temperature? And then you just get the right hormonal mix. Bang! Sweet as. You see this one? They put the whole mix in. MS salts plus vitamins. So if you that comes in one packet. MS plus vitamins and sucrose, then all you got to add is that little bit. Not one packet, depends how you buy it. I get mine from Portugal. I wanted to say that. So here's some more recipes. I'll just slowly go down. You can pause it later on. I think it ends pretty soon. Look at that. They reproduce the cotyledons. How cool is that? Holy mackerel, using cotyledons as explant, because I had on, what's that rad cultivar? It had an expression where it's a COT1 expression. It's a COT1 gene, because that's the gene involved in the cotyledons. It had a COT1 expression where it branched. It was so cool. It was a mutant branching. Righto, nodal segments, cotyledons. Oh my goodness, I didn't think there was this many. So you can see from these different explants, wow, from where you want to do it. That's cool. There you go. Wow, so medium composition. So yeah, that's what it'll look like, but in your Chinese dishes. And you know what I mean, the round plastic containers. I shouldn't say Chinese dishes, but that's what you get it in. PGRs, that's just the plant growth regulators, your hormones, because remember, it's a hormone if the plant makes it, it's a regulator if we add it. What else is interesting in this one? Protoplast isolation. Oh gosh, how cool is that? You can isolate the area of liquid of the cell. Ploidy. This isn't rad. Ploidy engineering. I haven't talked much about the ploidy. I've been doing a study for a couple of years with ploidy. It's going very well. Some pretty effects of polyploids. Is this going into the polyploid? It is too, which that's a bad example. I've got some really good results from polyploids and tetraploids. I've actually bred a triploid, which is hard to do, but it produced tetraploid offspring, which means that it was a successful chromosome transfer. So if I was to breed them out, I can get triploids with anything. Right, now what else is there too? Actually, what else? Anybody walking lightly? Nice to see you, mate. Very talented grower. Great for rooting and keeping the girls bushy. Yes, Raptor. Some really cool stuff. Do you want me to elaborate really on more tissue culture stuff? Oh, I haven't gone through the processes for before on what Kenna Corso was saying. I suppose I can touch on them, even though it is in the other. All right, I'll find my class. Oh, I haven't been through all those other. Okay. Look at me getting all flustered here. Unreal, isn't it? Yes, it is. I was going to go through my tissue culture personal one, just in case there's something in here that's interesting. Might help some people do theirs. That's just getting it ready, putting in a mixture. What part do I want to cut off? Keeping it there. This is what the genes that I want to take off. Now how do I want to culture it? That's a good example. So that's had too much oxen put in and started to root. See up top? The shoots coming out the side from the pericycle cells. Here's a bit of embryogenesis. So yes, look at that ovule. Multiplying that, and that's how it forms it, how it divides its cells. Different media. That's very cool. Look at that callus, that's so rad. See how they look all odd. Protoplast fusions. So when you mix, you can fuse cells of similar species is another way that you can do it. And you'd grind them up and you'd isolate and you'd put that in you denature the DNA and refuse them back together. Denature using polyethylene glycol. Synchron is the difference. Heteronote, I think that's very technical stuff. Microbeads. Oh, here you go, cool. Here's an old school recipe when they were trying to, so if you want to do a media, which is heavy, or indole abscisic acid is the oxen, and this is a kinetin. So if you've got ones that equal, this is an old school media. I wouldn't suggest doing this with cannabis, so I'm not going to even show it. But it was a formula where you could go off. At least it was something back in the day. It was pretty cool. I'll get on with that. Different lights on how they works. Yes, and red I don't like. In control organisms. Oh yeah. Here's some different media suggestions. You can find out if it works with you. yeah, there's a bit of, yeah. Wow, look at that. There you go. So MS basal medium with vitamins and sucrose, M5501. You mix 0.7 with 20 mils, it suffice. You can go up and start. Microspore culture, isolate the callus. So after you get, so you know the spores that you get from plants, from pollen grains, you can culture that as well. And instantly, that's homozygotic. So these chunks in here, all you do, see it's a little bit small, so you'd let it grow a little bit bigger, that's all right. Maybe a week longer, and you'd isolate, you cut it, subculture, change media. So I would get that, dip it into the sterilized, put it in my little laminar flow hood, which was my upside down tote, and subculture, like previously spoken about. So yeah, microspore culture, spores, double haploids. I'll go into that. That's through spore culture as well. There's some massive big explains. Oh, this is what I was looking for before. These are the little things you want to cut up. So see how they've just got so they had that big stem that I had before, then out of that, they'll go and cut them up even further and see, how do you know which stems to get? Well, look, right in the center there is the meristem, right where the nodes attach is the meristems. So just take that bit. So it's cool. So after this bit, you could use this, or you could just trim it off a smidge more, like over here, and it's sweet as. What's this one? Plant tissue analysis and tissue testing. Oh, that's how to test your tissue. We're not going to talk about that. Yeah, because it's important to test your tissues at different growth stages to see what's being absorbed and what the plant's signaling. If it's has it got any mutant genes? Is it all signaling pathways working correctly? Does it have any mutant traits that's exposing? This is a cool grow room, the tissue culture room. Different setups for different ideas. Comes back to there you go, the home one. That's that experiment. These are the cool bioreactor. The Raptor was trained before, so everything's just involved in one. It's all got at one. You just plug it in, put the next one in. It just all does its processing. Bioreactor. And automation. Remember before I was watching, put that video up that showed how the automative process works in the lab, how they do this bit. So then they've got to drop them out to here, then it goes on a conveyor, bang, into the truck. They're already doing it. There you go. There's the dishes. Perfect. Cheap as. Make sure that they're food grade though. They've got to sound at the bottom, the food grade, microwave safe. Go for the lights. This is all the lights. It's important to know you just don't need much lights. It doesn't, I think it's about, I get 1,000 lux and it suffice. Oh, that's right. This is the parts to do microspore cultures and haploid cultures and pollen cultures. Another way to do a cool culture is on a synthetic, on a microscope slide. You can do it and you can watch it grow. Done that before. So these are you can do the male bits, which is the pollens. Inside here, you've got pollen grains inside of the anthers or bananas. You can cut up the bract because they're all cells in there, especially the ovary in here. This will have parenchyma cells in here because it's ready to multiply when it gets the pollen grain comes down here and round through the micropyle in the end. So after it's going in there, it has the rad cells, so especially in there, which is down the bottom. So you wouldn't cut off the top bit, you cut the bottom of the bract or the calyx. Seen that? comparison, gone through that. Oh, the bioreactors, how they get the explants, spin them out, and they got different extractions that come out here for gases, because there's different gases that are involved with pathways for the plants that can stop them. If you've got too much ethylene, it will inhibit a lot of things and stop growth proceeding. Yeah, that's it. So that's my little one. Hey, cheers. Nice to see everybody. So does anybody want me to elaborate on the different types of techniques that you can do, like Kane Corso was talking about earlier? Or Kane Corso, do you want me to elaborate, mate, if you're still there? Because I've been going for a good hour. I don't mind, but I do everything for you people. So if you people don't, you folk don't really want, that'll be all right too. Trying to keep you happy. Plant cell, looking at my courses that I did, plant cell bioprocessing. Yeah, in here, there's got so many different ways to do cultures. So I can elaborate on that if you'd like. I suppose it is kind of relatable. If you don't like, you can best pretty much leave, eh? So why not? I'm going to go to the toilet. Factors affecting plant cell culture. So yeah, the explant source, that's a good point. I didn't actually go, if you've got mutant genetics or if you've got fungal diseases or problems in it, yes, you're going to have problems too. Composition, yes, a problem in everything. Aseptic conditions is a mega must. It'll fail. This is probably, I reckon the most important is aseptic, because you grow, where's that chart? Oh, well, you can multiply microbes 10 times faster, 100 times faster than you can plant cells. Right conditions and genetics. I'm going to say skim. Development of suspension cell culture. I'm going to get into the technical stuff. Cambium layer, no. Sterilization. Where's all the different microcultures? Here's the one on the microscope slide. So you put cover glasses down because you want to lay it in the center, and then you put a bit of cover glass on the top to cover it off, and then you put paraffin oil because you've got to seal it off from like getting fungus and stuff in the growth. Paraffin oil, or what do I use? I think Vaseline, and then it just grows under it and you can watch it. So that's zoomed in on what it looks like. So you just have the cells under there in your growth media with the cover slip over the top, and see there's a little bit of hollow underneath it. That's where it can do its thing. It's pretty cool. Growth, plant cell viability. Yeah, this is another thing if you want to get into it. If you've got a compound microscope like I've got, and I went and got a stain kit, and you can add these stains and just to see how viable different parts of the plants are. You can do bioassays. Applications. Oh, yeah, righto. Technical stuff I'm looking for. Natural nutritional requirements of a plant cell. Remember, it was 5 things. Inorganic sources, carbon, vitamins, your hormones, and organic supplements. So, Murasagi Skoog medium and hormones. And then they alter at different amounts depending on what genotype you got. Right, now it goes into the different types of ones, auxins and growth regulators. No, come on, where's some other different cool cultures? Organogenesis, somatic embryogenesis, or somatic. Where's that get to? Types of differentiation. Just trying to skim through to the most interesting bits, because I've already gone through all this stuff in detail in the previous tissue culture lives. If you'd like to look through there, somoclonal variation. That's just well, somoclonal means like gene wandering or mutant type things. Factors affecting, yep. I've been through all this micropropagation. Here we go. There's some interesting stuff to share. Yeah, while we're here, these are little growth stages. So if you're doing embryogenesis, where you're doing pollens or spores or those types of things, it'll go through these 4 stages. And that's it, distinctively goes through these 4 stages. The globular, looks like a big round glob, then it'll form a heart, then it'll go long and skinny to a torpedo where it's starting to get its shoots up the top, organized, and then it'll start to throw out the cotyledons. Protoplast culture, I suppose. Well, that's a bit interesting. It's the layer around the plasma membrane of plant cells is surrounded by cellulose wall, and the adjacent cells are adjoined with thick pectin-rich matrix. That matrix is a protoplast, and that's the stuff that you can culture as well. Isolate. I'm not going to go into the details because I'm just going to really talk about the stuff that you can do because they're really interesting. And if you want to go into details, you can ask, and I can. Or I can, you can pause these. Synthetic seeds, these are so cool. I was amazed. They're so rigid. You can't squash them. Yeah, cryopreservation is very important. If you want to preserve your genetics, germplasm is gene preservation. Isn't that a spin out? How are they different? Secondary metabolites. Yes, that's what I am. That's one of my main roles is secondary metabolites in plants. I try to manipulate their pathways to try and express more terpenes and flavonoids and cannabinoids. This goes way out of tissue culture. I'm not going into that other stuff. Sweet. I see Raptor said something. Great presentation, Aussie. Thank you, Raptor. Appreciate your good feedback, mate. All right, he's giving me props. I'll keep going. I'll see if there's anything in this week 5. Precursors. Is that tissue? Not really. Elicitors. No, this isn't. This is how to program cells or how to work with cells to make them alter or understand them or see what state they're in or change them. This is what this start precursors and this is anyway. So that's it for that tissue culture bit, I think. What was these interesting things I'll put under here? Where's no? Oh no, that's not tissue culture. Right, I'll see if there's anything written. Great presentation. Appreciate your good feedback. I'll see if I've got anything else, mate, that's related. What's my other culture classes? Mushroom, cell biology, no. Plant biochemistry, yeah, and biotechnology. Yeah, this one. Plant biochemistry. That doesn't talk about tissue culture much. I'm just going through it. I'm not sharing. But biochemistry does. I'm sure it'll go through it. That was a pretty cool course, that. What? Oh, yeah, I got heaps on it. Heaps of stuff to share. Okay, I'll get back up to it and share the screen again. Oh, Raptor. I'll just get rid of that. I appreciate your nice feedback, though. Thank you, mate. So here, these are the lectures, and you can see embryo culture, callus culture, single cell culture, protoplast isolation, factors affecting secondary vito, hairy root culture, some clonal variation, synthetic seeds, germplasm conserving genes, direct methods to transfer genes, transgenic plants, morphogenical and DNA markers, microcystic selection. Okay. Requirements for plant tissue culture, micropropagation, PRACT. Oh yeah, I'll go through the PRAC for you. Callus induction, it's a PRAC again. Special analysis of DNA, cell suspension culture. Determination of molecular size of DNA sample. Oh, let's refer, no. DNA, no. Okay, so there's a few ones here that are related. Like the hairy root culture is pretty good. No, actually, no, I'm going to go with the, I like micro-assisted selection stuff because that's how to do stuff, how to get what you want. Requirements for a plant cell culture lab. Let's see what's written here. So it's really just been sterility. Well, that says the equipment there. See, from now that I've gone through all this stuff, you can realize that you don't need all these things. If you're going to do it commercially and produce for other people and sell them, yes. But for doing experiments for the average person at home, no. You can get away with it with a lot of cool things that we've already been through. So they're the equipments that they say and the materials. So it's just really the MS medium and some bottles. Yeah, so they've covered it too. There's some how to prepare it, how to make up solutions. Yes. I was hoping there's some cool slides or something that glossary. So I mean, it's the end. These are all right. Here's some frequently asked questions. Right, the requirements for plant cell. No. What are the main components of the media? So I've been through that extensively, I think. The various sterilization techniques used are through the autoclaving, filtration, dry heat, laminar airflow cabinets, irradiation using your UVC light or UVB actually does have some effect with microbes too, which we saw before with those lights effects, and surface sterilization. So that's massively important. Sweet. Now, is there another one? Let's show another one. Micropropagation or here, yeah, we'll go through a PRAC. PRACs are very hard to find. Oh, they were back when I micropropagation PRAC. So you can read all this if you're really interested. You can pause it and stuff, because I'm definitely not going to go through what the PRAC is because I've kind of already summarized it, but I'm just showing different techniques from people. This is a university written course. So this is the way that they have been instructed to do it and the way that they've had success. So there you go. So it just shows step by step exactly what to do and what to add. Really, is it written by her? That's my teacher. Collect, rinse, sterilize equipment, sterilize, keep sterilizing way more because they sterilize all their equipment more. So cotton plug culture, okay, and then mix in the medium. Make sure your medium's good with the right temperatures and for duration. And the hormones have gone in after a few weeks. It's good. And then subculture for another few weeks. And then you can harden up. How long do they harden up for? Bags having a few holes in them for initial 10 days for hardening. Harden up for 10 days, eh? Okay. I reckon, all right. I suppose it depends what type of sun you're using or types of lights it's going into, but 10 days you're on the safe side. You're going to get no deaths. I would probably go maybe nearly half that, knowing me, or 7 days. There's another PRAC that you can, another PRAC. Let's go through another PRAC. How about a callus induction practical? So to start is callus induction and plant rejuvenation. All right, very good. Theory, no, been through it all. We want to multiply callus. Procedure. So it's the same again. Rinse. They're using leaves as the next plant. So you can rinse your bit off, put it in detergent again. And the only difference from the callus is the media, that they've used more of an equal oxygen to cytokinins ratio media. So it won't lean either side, it'll form the callus. It won't heavily weight from one hormone to make roots or heavily weight from the other to make the shoots. So there you go, and hardened off again. So there's a little way. They suggest here to use a metal. I don't suggest you use that at all, one bit. They should be only done in in lab conditions. And the reason being is because you've got to get rid of this. To get rid of these heavy metals is hard. If you put it in anything, it's going to absorb into it. And if you throw it in the bin, well, that's just damn terrible. There's ways that you get them to be removed safely so they'll bind with other reactants to make them not dangerous. So I really don't, just because it says it here, don't do it, please. These are only just different ways that they've done it, and these are lab techniques. So I really would like to promote the safe techniques and usings of everything that I get involved with, because it can go dangerous quick. It's one reason why I haven't talked much about ploidy or polyploids, because there's some dangerous practices in that if it's not done the right way, that will go bad. Actually, speaking of that, I didn't do the di-haploidization. This is another way that you can multiply cells fast. So you've got your, no, I'm not going to go that one. A bad example. I want to show where the pollen culture is. Here we go. Is that it? Yeah. So here's where you got your spores. You put your microspore out. It'll go through its mitosis. It'll bind. It'll go again, and it'll start and transfer, and it'll start and rejuvenate. Sorry, it'll start and generate more cells, which will multiply of itself. So you'll get a direct copy of what itself is by doing it this way. And then straight away, you've got a very homozygotic plant or very stable genetics. So for those people who have gone and bred a lot, they just have to go and do this after with their F1, and it'll really stabilize it a lot. It should eliminate a lot of things. I'll go through this a bit. It's very interesting. It's very technical. It's of getting pollen grains and multiplying them. So I can store my pollen grains successfully for up to 5 years. I've used them and have them reproduce. Yeah, so stored well. It's a good way for your germplasm or your genes to be retained, because remember, seeds are only generally about 12 months you get out of them, or up to 3 years in good conditions, or you have to really do some sort of rescue after that to try and bring them back through tissue culture is a good way. Haploids, it's technical again. Anthiculture. So here's the anthiculture where they've done like we were talking about before, just under the microscope, and they've used the blue METHY stain. Anthiculture. So see how it's growing a callus inside of the anther in there? So that's the long banana that they've split open, and then they just had all the grains inside of it. So instead of putting an actual pollen grain out, they've done the whole anthiculture, or banana culture, as cannabis people know. Embryogenic calluses, yes. And anthiculture, there's another one, just its different growth stages, how it goes through. So it starts to get its callus growth, and then it starts to get chunky. Look at that. I love those. The callus looks really odd when it grows. It's cool ads. You can touch it. It's sort of solid. It's yeah. But anyway, you shouldn't touch it. Heterogenic, yeah, because too much cannabis has a heterogenic population where it's got too much mixed genes in it, and that's why you get your unstable genetics and your problems. So by doing it this way, you can make it homozygotic again and make it your F1 polyhybrid into a very stable genetic line. And it's fun too. Isolated microspore culture in barley. See, a lot of these things that I get, my science that I'm not taught directly in cannabis, because that, as far as I know, doesn't exist. So I do agricultural science and plant soil science and microbiology for agriculture, and that's Then I link all these things that I learned back into cannabis, and that's how I get all my research and my experiments from that. And that's why some of my experiments that I'm doing haven't been done in cannabis yet, because it just hasn't been introduced in the area. People need funding to do all their experiments, and it's the home blokes or the home females, they generally don't do a lot of it because they're just happy with where they're at. I'm interested into how to get more out of it and create better things. Embryogenesis in microspore culture. Very cool. So I hope from this you've really picked up that it's all about getting the right sort of material at the start, sterilizing everything so you reproduce only plant cells, and then putting the right hormones in to getting what you want at the end. You can do that with anybody, can do this, even in flower. So the advantages of this, if it's flowering and it's stinking bad, it's chunky, it's finishing early, all these traits that you like, you can go and grab some nodes off it and culture them. So instead of having to put it through a regenerative cycle afterwards and taking up space, you can go into the lab and just go in your little lab area and get succession over that. And then also probably maybe ridding any viruses or making it cleaner than what it originally was. This is a nurse culture. That was one of my exam questions I got wrong. Let's see answers again. Morphological differences between haploid and diploids. Haploid is when it halves the original cells and you breed with that. So if you would, what that means is this plant put out pollen, which was a diploid, and then it got halved, and then the haploid was reproduced. Sorry, that cell was reproduced, and the outcome was the haploid. So it's just probably showing, I reckon it would be the better one, because it would have eliminated all of the unfavorable traits from the second set of chromosomes. Okay. Induction centrifuge. This is another way that you can isolate cells by putting it through a little centrifuge. Excuse me. Microspore culture. This is also why I'd like people to help contribute, or I don't beg for money, but money's nice, and because I need all these different tools and techniques that I can't afford. There's so many different ones that I'd like to test that I can't test for, etc. I don't like begging or asking for money though. Showed that one before. Cells, all right, I think we're getting nearly to the end. I haven't gone through any real details. Induction of in vitro culture of the ovules. Oh yeah, this is, remember I was saying before with the bracts that we've got, if you can get the little bottom section of the bottom, that's a really good way to be multiplied too. This is just showing it. Sterilize them, pull them out, sterilize them, seal it up, and wait for little callus chunks to grow. Oh yeah, the parts, different lights, experiments to see which ones were better. Cultivar dependent. See how cool the chunks of callus are? Look at that. Globule blobs. Stages. Okay. Tetraploid cross. Why is this in here? If you cross 4 and haploids back to 4 again, because it'll go from 4 back to 2, and 2 plus 2 is 4. So that's why with my tetraploid, I can cross that with a diploid, so it'll halve, it'll go from 2 to 1, outploid will be 3, a triploid. And I got 21 seeds of that, and they should be worth thousands of dollars each, because anyway, they're only going to last for a few years too. They're only probably 12 months old, because I've tested 2 of them. I have to try and respect the law, plant counts. This should be employed. This is a difference. If you have extra sets of chromosomes, it's a different set of genes, so to speak, to handle situations. So you usually get better outcomes. It has dependent, more ploidy has like better disease resistance as well. It's got a set of genes going, oh, well, I'll just do something. What do I do? Oh, I'll just take care of this for you, and can activate different pathways and stuff quicker. Less ploidy difference, one of the advantages. Reading techniques. Okay, that'll do. What do you reckon? Probably going for way longer than an hour. Is there any other questions? Get them in there right now, please. I can have, how do I put me back into it? That one. Hello. Yeah, there it is. If you've got any questions, please ask them now. Are you kidding? It's nearly 2 hours. Holy mackerel. Wow. It's so fun. And this is things that everybody can do really, really easily. And then let me know how they go. Also, if anybody's interested in, I run a thing on Zoom, a channel called Weed Nerds. If you're interested in experiments or doing experiments, please email weednerds@protonmail.com and tell me why you'd like to join. And you might be joined. And it's for people that like to do experiments. So we share our experiments. We get opinions of other people and help out each other in that sense. Yeah, thought I'd mention that too. So that's weednerds@protonmail.com. And just let me know why you'd like to join or how you'd be, yeah, just why you'd like to join, really. And it's got nothing to do, I respect the law. So if you think you're going to get in and get cheap or free or blah, blah, that's not what it's there for, because I respect the law. I hope you do too. The world, the things are changing now. So, it can get better. We don't have to go like we used to do or whatever used to happen. So, yes, it is changing for the better finally. So it's good. Good on you, Dave. Appreciate that, mate. Raptor. Dave says, good show, good vibes. Thanks, mate. That's what I try to bring along and try to help out. No drama. Just talk about the plant and how it can bring the best for us, because we've all got our own blimmin' problems. So we don't need no nothing. Today's the 1 year anniversary of the show. So I'd like to thank everybody too for contributing and helping and keeping me here. And it's mainly you folk in chat like Dave 1969 that are doing that. And Raptor Grow. Cool folk. So what's Raptor say? All my presentations have been 1 to 2 hours and I barely cover the basics. It's such a fun and fascinating subject. Yes. Yes, it is. As long as we keep doing all of our experiments and sharing our experiments, things will get easier for us, because they make out, if you watch some other programs, they suggest they make it too technical, and plant science doesn't have to be that technical. You can go and use all these big names and work out pathways without doing, and you can do successful morphogenic breeding really, really well without getting that technical, without getting the heavy equipment and all that sort of stuff that's needed. So that's where I just like to break it down and make it easy for everybody so they can do the fun stuff too and enjoy and get the best out of their cultivars. Because if you find some stuff that agrees with your body, it's, I don't know about you, but in my experience with the thousands probably of different cultivars I've smoked, there's only a handful that make me feel good, that don't get me too anxious, that don't make me too hungry, that don't put me to sleep at the wrong times, etc., etc. So by getting those good genetics and maintaining that, it's good for the person. And tissue culture is a rad way for that to happen. So how I could keep my genes going, here's a good conclusion. How can I keep my genes going for 20 years? Well, you really can't. Yes, you can. Here's how to do it. So I've got pollen. Each keeper that I find, I'll make pollen out of that. I'll induce it with silver and produce pollen, and I'll store the pollen successfully in its ideal conditions. And I've done that for up to 5 years. I've stored it. So out of that pollen, then I can go and put it through a culture and reproduce that plant and do the same thing again. So every 5 years, I could just do the same thing, and I've guaranteed those genes have maintained for 20 years. That's one example. It's really cool, tissue culture. And then you can work, I didn't even go into secondary metabolites. So you can start, and do marker-assisted selection in tissue culture, where you were trying to get different outcomes from different Petri dishes. So in this one, you might put in too much of this chemical or too much of this hormone or something like that, or to change its ways, like you use an elicitor to do that. And that's cool ways to manipulate things. So all these different fun things, just anyway, I find them fun. So if you want more fun, yeah. Anyway, thanks very much. I think I've talked enough. I really appreciate everybody for your efforts in chat. Thanks again. Next week, what are we talking about next week? Yeah, I'll be here next week. Definitely. I've already prepared it. I've prepared the next few weeks in advance. Next week is what terpenes, is it really? What terpenes increase the CB1's receptor activation or the entourage effect? So it's a big terpene special. Do you want a sneak peek? All right, I heard you. I bloomin' heard you. Pollen, here you go. So I haven't got many slides yet, but I've got this one study that is amazing. That is just amazing. So I've got a few different things that, and I'll talk on terpenes as well. I might elaborate on that. And how cool! My next class this semester. What's the name of it? One of my classes this semester is on terpenes. It's oh no, I'm not sharing. Let's have a look. Hey, good on you, brother. Thanks, mate. Yeah, anyway, I'm not going to, it doesn't matter about the title of the class, but yeah, this semester it starts in 3 weeks' time. It's a terpene class. So that's going to be something cool that I can really share as well. So yeah, next week. Raptor says, thanks for a year of knowledge, being busy with family and school, but I watch when I can. Yeah, I know, mate. You're a good bloke. I've always enjoyed your company. Thanks, Raptor. He does lives too, everybody, if you want to get involved. He's and he's not that one that talks out of experience levels or tries to talk where it's not. He talks from experience, and his information is quite good. So he's yeah, like myself. I don't talk out of school type thing. I like just to talk about from what I've done, my experience, and what I know through my studies, and then I can back it up through my slides. So I don't like to, if I don't know something, I'd just like to say I don't know. It's a lot easier. Then people can try to trust the words, because I like my things to be fact checked. Thanks for walking lightly. Terp on, dude. Yeah, that's what it'll be next week. The terps will be on. And I tell you, there's another bonus from doing, this is amazing. This is some really, really full-on stuff. I'm playing with ploidy, and I'm doubling the chromosomes. I've got a cultivar that is called Tetra. They're the one, she has 4 sets of chromosomes in her. And when I flower her out, tetraploid's supposed to have a enhanced terpene profile, especially in the sesquiterpene terpenes with the 10 isoprene cannon carbon chain. And this one is rad. It lasts, and it's probably 6 or 7 months old now, and it still smells. I'd still give it a 1.5, maybe 1.3% terp profile. It's still quite loud. It's very good. It's probably the longest lasting terp profile that I've ever seen. So I'll elaborate a bit more about that next week on how to get the best out of the terps too. Woohoo! Good, clean cannabis for everybody. Okay, thanks everyone. I'll sign out. See you next week, I hope, or through the week. If not, be member on the Weed Nerds at protonmail.com if you'd like to join that legal channel. Thanks everybody. Happy growing, happy breeding, and good health to you all. Bye bye.